Hello, everyone. Thank you very much for joining us for our February research webinar. My name is Alexander Maxon from the Huntington Society of Canada. We're thrilled to have you all here with us today as we learn more about promising developments in the search for effective treatments for Huntington disease, a condition that affects so many Canadian families and communities. For our webinar today, it'll be presented by Skyhawk Therapeutics for an overview of SKY-0515, including insights from their phase 1 clinical trial results and a discussion of what these findings may mean for Canadians. This is a very exciting opportunity to hear directly from the experts at Skyhawk Therapeutics about their investigational therapy, which is a novel small molecule RNA splicing modulator and designed to reduce levels of mutant Huntington protein and PMS1 in order to address key drivers of Huntington disease pathology. So just to introduce our speakers, I'm very pleased to welcome Kathleen McCarthy, co-founder and distinguished senior scientist at Skyhawk Therapeutics. Kathleen is a leading expert in developing small molecules that target RNA splicing. She has more than 15 years of experience in the discovery and development of small molecules targeting RNA, including the first approved small molecule to modulate RNA splicing, Evrysdi, for the treatment of spinal muscular atrophy. Kathleen originally began her career by co-developing a treatment for SMA as a preclinical scientist at the SMA Foundation. In that role, she helped bring the first ever small molecule therapeutic driving mRNA alternative splicing to clinical trials. The drug candidate was acquired by Roche in 2011. Kathleen moved to Roche to work as a preclinical scientist. She first and last authored critical papers in the field, including the Science paper in 2014, where the SMA small molecule targeting RNA splicing was heralded as first-in-class small molecule therapeutic that specifically corrects exon skipping, and the Nature Communications paper describing the SMA compound mechanism in 2017. Prior to her work in drug discovery and development, Kathleen completed a Fulbright Fellowship at Swiss Federal Institute of Technology in Zurich and graduated with honors in chemistry from Wellesley College. Joining her for this presentation is Penny Trefillis, Vice President and Head of Medical Affairs at Skyhawk Therapeutics. Dr. Trefillis brings extensive experience in rare disease drug development, medical affairs strategy, and a deep commitment to translating scientific advances into meaningful benefits for patients. Together, they will provide an overview of SKY-0515, share insights from their phase 1 clinical trials, and discuss these implications for the Canadian Huntington disease community as research progresses towards later stage trials like the ongoing phase 2/3 Falcon HD study. Please note that SKY-0515 is an investigational therapy at the moment, so it's currently only available through clinical trials and not yet approved for use. I encourage each of you to submit any questions you may have throughout the presentation via the chat function or specifically into the Q&A. We'll aim to address as many of them as possible during the Q&A portion that we're going to have at the end. So without further ado, let's turn it over to Kathleen and Penny. Thank you very much for being with us today. So over to you two.
Thank you, Alexander. It's a pleasure to be here. We really appreciate your time and the association's time you're giving us to explain a little bit more about Skyhawk Therapeutics and our molecule SKY-0515, which is currently in the clinic for Huntington's disease. Today, I'm going to speak about the SKY-0515 molecule, a little bit about how it's potentially going to work and its mechanism of action, and then I'm going to go into the clinical development of the molecule and where it is right now in clinical trials. So just as a background, these are the types of therapies being developed right now for the Huntington's disease. It's a really exciting time in the Huntington's field in general, and there are small molecules being developed, which Skyhawk has a small molecule, for example. These are usually lab-made compounds taken as a pill, and they may distribute widely through the body and can be designed to reach the brain. Skyhawk's molecule is designed to reach the brain. Nucleic acids are medium-sized lab-made molecules. They are also being used to target in clinical trials for Huntington's. They must be given by injection into the blood, or in this case, into the brain or the spinal cord for CNS diseases. and they tend to not spread as easily through the body due to their size and other reasons. Biologics are larger than nucleic acids and small molecules, and they similarly must be given by injection to the brain or the tissue of interest, and also may not spread as easily through the body. So those just an overview generally of where Skyhawk sits with other types of treatments. This SKY-0515 is a small molecule. It's designed to modify how the body expresses the Huntington's gene through the RNA. It's a once-a-day pill taken by mouth. Our aim is to slow the progression of Huntington's disease and potentially to reduce the levels of harmful proteins like the mutant Huntington protein and PMS1, which are both linked to the disease. It is shown to be well tolerated so far in clinical trials based on a 9-month interim data read from an ongoing phase 1 Part C trial we have in Huntington's disease. So our molecules are designed to fix or adjust how the cells function by changing the expression of RNA and thereby targeting the proteins through the RNA. So as you already know, Huntington's disease occurs because of an abnormally long CAG repeat in the DNA of a gene called HTT. People inherit this CAG repeat, which is CAG repeats are normal in the DNA, but when they're too long, they can have very consequences that are disease-causing. So this long CAG repeat sequence in the DNA causes the body to make an abnormal protein, the mutant Huntington protein, or mHTT. This mutant Huntington protein can then go on to damage brain cells over time. And then the effect over time is damage to brain cells can lead to brain changes in some regions, affecting mood, thinking, and movement in Huntington's patients. So this is pretty well known and understood, and an emerging separate cause of the disease is the somatic CAG expansion, which can occur over the lifetime in a patient. So patients are born with extra CAG repeats, and we know that the more CAG repeats a person has at birth can be linked to, associated with potentially an earlier age of onset of the disease. What's more of an emerging part of the story is the somatic expansion that occurs in the DNA of HTT, and this occurs over the lifetime of a patient. And it generally repeats in certain types of cells, expands. The rate of expansion can change depending on the cell type. So in brain cells, the rate of expansion, and in particular type of brain cells, the rate of expansion is faster, and that rate of expansion can affect and impact the onset and the progression of symptoms as well. This is a paper published in 2025 showing that certain cells in the brain have kind of faster rates of expansion. What's now emerging more and more is this role of PMS1 in terms of delaying or slowing that expansion through what it does to the DNA. So there's been some genetic studies which have shown that patients where there's also a co-mutation in the PMS1 locus or the PMS1 gene can correlate and modify the age of onset of the patients. So reducing PMS1 is a way of slowing the progression. It's potentially a way of slowing the progression of the disease as well. So SKY-0515 is designed with a dual mechanism of action. Our molecules target the mutant HTT and also target the PMS1 protein. And we target that through lowering the amounts of mutant HTT protein and potentially PMS1 to help that DNA repeat expansion over time and also reduce the mutant Huntington protein levels. And how the molecule does this is through a process called splicing. And RNA splicing, as you can see in the cell, the DNA is stored in a protective area of the cell, the nucleus, for important reasons. So we can keep the DNA from exposure that could cause errors in the DNA. And that DNA needs to get to another part of the cell in order to form proteins, which are the building blocks of the cells. So DNA is converted into RNA, and that RNA is a message from the nucleus to actually physically going to where the proteins are made in the cell. So RNA is the messenger, and that's why it's called messenger RNA. So the DNA gets copied into RNA, and the RNA then gets made into proteins. And our molecules affect right at this pre-mRNA, which is still needs to be edited to form the final RNA to be made into proteins. So from the DNA, we have the HTT pre-mRNA, which is copied and made. That needs to be edited to the final version. We need to cut out some parts of the RNA that aren't useful for the final message to get the final HTT mRNA, which is then converted into protein. In the event that we have a mutation, or in this case, CAG repeat expansion in a person with mutant HTT, where they have a CAG expanded region, that would be then converted into a protein that has aggregates, and those aggregates, that long extended region, can cause brain cell damage and then damage to the brain in general. So our molecule actually acts here in the pre-mRNA message, where we insert this little additional message or code, which can, when inserted, flag this RNA for degradation. So it's like changing the message to say, don't, make this protein actually degrade this protein entirely. And that's, we insert an extra exon, and that exon basically gets degraded. The whole message gets degraded, and no toxic mutant HTT is formed. So Skyhawk 0515 is a daily pill designed to potentially slow or reduce the damage caused by HTT. It's taken by mouth once a day. The pill can spread throughout the body, and including the brain. And as I mentioned, it's designed to lower the amounts of the HTT protein, which could help prevent the brain cell damage, along with designed to knock down another protein, PMS1, through the same type of mechanism to reduce the amount of CAG repeat extension that's happening in the cells. So if there are no questions there, I will move into the Skyhawk clinical development. It's for holding questions to the end. So this is the standard clinical development program for most biotechs or pharma. They may follow the same process where you have preclinical data, where the company or the pharma people are analyzing the molecules in the lab, in cell-based systems, in animal models of the disease. And once they think that they have, enough safety data and also data on the efficaciousness of the drug in these models, they'll apply to governing bodies to go into the first phase of clinical development, which is safety studies. So phase 1 is a small cohort of usually healthy volunteers or sometimes patients, where we study the safety of the drug for a certain amount of time. And we can also get some more information about early signals of efficacy, early signals of biomarkers that may be important. And once that phase is passed, we then again apply to go into a phase 2 study with regulatory bodies as well. And in phase 2 studies, which is, you can see where Skyhawk is now, we look at safety again in a larger cohort, and this generally is people with the disease that we look at. And we'll look at different doses, and we'll look at the efficacy and establish early signs that the drug may be working through biomarkers and clinical assessments. Phase 3 is even a larger cohort of patients to confirm again that safety and also confirm efficacy. We need to show statistically meaningful results in order to be able to go for approval country by country. Right now, Skyhawk is conducting an ongoing phase 1C in Huntington's patients in Australia, New Zealand, and a phase 2/3 in Australia and New Zealand. And we are progressing with our global phase 2/3 study, which is started in Georgia and is starting to progress across different countries. So, as I mentioned, Skyhawk is currently running a phase 1C study. Again, a safety study with maybe some early looks at some type of efficacy and biomarker readings. This is the structure of the study. You can see that the first 3 months of this study have been completed, where patients have taken either a low dose or higher dose of the drug along with a placebo those first 3 months in a small group of patients. Some of the inclusion-exclusion criteria of which patients we enrolled in the trial is here on the left-hand side, where we have the ages of the patients ranging from 25 to 70 years old, the stage of the disease. We were looking at early to mid-stage disease with these types of scores on some of the clinical outcome measures. So TMS greater than 6, IS greater than 70, and TFC greater than 8, greater or equal to. So that part has been completed the first 3 months, and we have an ongoing extension of this study in the patients where they were assigned to either a low or high dose. So we no longer have the placebo, as you can see here, and patients are ongoing. They're currently in this 12-month extension period. So the main study goal of this study was to check if Skyhawk SKY-0515 is safe and well tolerated. Additional study goals we had were how it was distributed through the body, so it's PK in the body and the brain, how much it lowers HTT and other related markers in the blood and spinal fluid, MRI looks, brain changes over time, and the outcome measures related to movement and thinking tests are also studied. And these are some of the results that we've published in our press releases over the last year or so. We see that SKY-0515 has lowered mutant huntingtin protein in HD patient blood over this approximate 3-month period. So on the y-axis, you have the amount of reduction in percentage relative to you can see the placebo at the top. So these are patients that did not get the active drug. They got a non-active, a placebo, which basically we have to have in order to show meaningful effect. And you can see the 2 different doses of the drug at 3 milligrams or 9 milligrams. This was a small study again, but you can see that there's a reduction of about 29% of HTT protein and mutant HTT protein in the blood at the 3 milligram dose, so the lower dose, and at the high dose, there's a 62% reduction in mutant huntingtin. These patients, again, now have all been transferred to either dose of the drug, and now they are being evaluated in a longer we're now through 9 months. And the other press release we released was about our 9 months data, actually on the CUHDRS score. This is a clinical outcome measure that's used widely across the Huntington field. And generally, we're looking at progression over the course of a year or more. This is an interim look in a subset of 17 patients. And we've pooled the 4 milligram or 9 milligram dose of this study. So you can see how patients have done over these 9 months in blue, where you see an increase in the CUHDRS of about 0.64 compared to a natural history control. And natural history control is using some of the data that's been made available by different CHDI, for example, that have been doing these natural history studies where they're looking at patients over time without treatment. And so it's our molecule compared to a natural history control. So it's not patients that were in this trial in particular, but it's looking at patients that are representative of the patients we have in our trial, and we're doing that comparison. So that is, again, this is early phase 1 data in patients, and we're looking at their scores over 3, 6, or 9 months. And we're encouraged by these results, but we also know that this is a really early look. And our aim for the molecule is to, most of the aims of therapeutics in this area is to slow the progression of the disease. So we still have to look at the molecule in larger phase 2-3 studies, and I'll talk about those studies which are ongoing or planned in just after this slide. So just to sum up the part 1 phase, phase 1 part C interim study results, we've seen good tolerability, so the drug looks to be well tolerated by patients. No significant signs of harmful side effects have been identified across all the explored doses. Our PK, what we've seen so far is we've seen a dose-dependent increase in the amount of drug based on dose level. So we see that nice proportionality of our drug reaching stable levels in about a week. And that it's been measured similar levels of drug in both the blood and CSF. So we think we're getting into the brain effectively. And then the interim results clinically, we've seen the biomarker changes. We've seen a lowering of HTT mRNA and also protein by approximately 62% in the blood. And then also we've looked at PMS mRNA lowering, and we've also seen lowering there about 26%. And then the encouraging trends in the CUHDRS scores over time, showing the potential to slow disease progression relative to natural history. So we're excited about moving to the next phase, and I just want to pull up our phase 2-3 Falcon HD study, which is ongoing in Australia and New Zealand and opening up in our worldwide locations. This is the general view of what our study looks like. These are 2 different studies. So the Australia and New Zealand is currently enrolling patients. We'll look at the primary outcome will be a measure of mutant huntingtin in the blood over 12 months. We have a low, mid, and high dose and placebo for this group, and participants will be randomized into receiving either the drug or placebo, where neither the patient nor the doctor knows which drug they'll be getting. Our very general overview of the inclusion-exclusion criteria, we're looking for age 25 and up, HD gene expansion of CAG of greater or equal to 40, and more in the early to mid stage of disease. Additional study goals are here. We're going to look at safety. We're always looking at safety all the time, any serious side effects, measuring brain changes in terms of sizes of different areas using the techniques developed in the Huntington field, the size of a brain area region called the caudate. We're tracking movement changes, thinking, and daily function on a consistent time frame, and to see if there's some slowing down of or delay of HD progression. And then the over worldwide study will be looking over 18 months with the same type of doses and placebo setup, and our primary outcome are clinical outcome measures. So this is currently the inclusion-exclusion criteria of the Falcon HD study. I just wanted to point this out, that it's not only those things we mentioned on the previous page, but there's a long list of inclusion-exclusion criteria that the principal investigator of the site or neurologist will be able to evaluate whether a certain individual is fit for this trial. That's independent of the sponsor. So this is a question we get pretty often, and we just want to talk about patient access for the drug after the trials. Obviously, access for non-participants of the trials depends on several factors, which are not limited to the overall study results, safety findings, regular decisions in each country, and then the development of the procedures and how the molecule can be distributed. Along with that, there's an open label extension studies that potentially get opened, like in our phase 1C, where we offer the drug to trial participants after the main study ends. And those tend to be non-placebo controlled, so it tends to everyone gets switched onto drug. It just depends on the study results, really. And then we are very committed to timely access. We know this matters deeply, and we're working to move forward as responsibly and efficiently as possible in the clinical development process. Lastly, I want to talk about some study information. So both our Australia, New Zealand phase 2/3 study ongoing is available on clinicaltrials.gov to get more information, as well as our worldwide study here. And these are the identifiers, and we're happy to distribute them after this call as well. So we have a website, falconhd.com, that also provides more information, and any general questions or inquiries can go through our email address, which is SKY-0515 trials@skyhawktx.com. So thanks a lot for having us.
Just wanted to say thank you one last time for taking the time out of your days to come and speak to the community here in Canada. We really appreciate it. If you have any questions for me, feel free to send me an email. If any of the audience members have any additional questions that you would like to ask at a later point, you can also contact me at Huntington Society of Canada, and I'd be happy to pass on the questions, get answers if that's okay with them, and then share it again with the community. So thank you once more. And with that, I think we can end today's webinar. So I really appreciate you joining us today. Thank you.
Thank you so much. Nice to meet you all. Thanks a lot.
Indeed. Take care, everyone. Be well.
Have a great day.
You too.